All right, Cross Hill 8th graders, welcome back. So I'm now set up to do the chemical indicators test, or the chemical testing, which is on page 3 of your lab packet. As you can see, I have my 6 test tubes set up here and labeled. I've got water, glucose, and starch. And then I have our starch indicator solution, or our iodine. And then I have our glucose indicator solution, also known as Benedict's solution. Another word you guys need to memorize, Benedict's solution. So when you see them talking about a blue indicator or glucose solution or Benedict solution, we're talking about this lab. Benedict solution, glucose indicator. All right. So we're going to start with, because the Benedict solution, for that to work, it needs a little bit of heat. So I'm waiting for my water to boil over here. So we'll get started with the glucose. So basically what I'm going to do here is I'm going to put a few drops of glucose in each of these first 3 test tubes. So 1 water, 1 glucose, and 1 starch. And then we're going to see if anything happens. All right. Ooh, oh my goodness. Okay. All right, so here is my starch indicator solution, or my iodine. So I'm going to put that into our water. And our glucose. And then into our starch. I'm going to stir each of these up a little bit here. So this is our starch indicator. Here's your glucose. And here's water. So we noticed a color change in one of these test tubes here. And it happens to be in our starch test tube. Our color change is a little hard to see. Let me see if I can get it under the light here for you. Not really any better. All right. So what happened here is it turned our starch indicator, our iodine, turned our starch solution, right, our starch test tube, into a blue or black color. And that is how we know that there is starch present in a solution. So we're going to go down here. I'm going to move this back for a second here. We're going to go down here to our chart. And we started with our amber colored starch solution. You can also put in here parentheses iodine, right? Because there's lots of names for these things. I want you guys to make sure that you understand that they all mean the same thing. So we started with our iodine. We tested our distilled water. And what color is our distilled water? Amber, right? I told you to remember that color, amber. So I'm going to write amber in that box, right? Because we had our color change to amber. I'm going to skip over here to our glucose box. If we look here in our glucose test tube, did we have a color change? We did not, right? Glucose test tube, we had an amber color present. So we're going to write amber. Okay? And then our starch test tube, we did have a color change. And you're going to write blue-black, right? Okay. So your chart for your iodine solution should look like this. For distilled water, it should be an amber color. For starch, it's blue-black. And for glucose, it's amber. And the reason being is that there was no starch in the distilled water, and there was no starch in the glucose. However, there was starch in the starch, right? And that's how we got that color change. All right. So it seems like my water is almost boiled here, ready for our Benedict solution, or our glucose indicator. So what I'm going to do here is I'm going to add my Benedict solution to my 3 test tubes right here, the 3 we have not used yet. And then we're going to, because Benedict solution needs some heat to work, I'm going to heat it up for a minute. All right, so I'm running out of clean pipettes. All right. So here's my Benedict solution in my water. My Benedict solution in my glucose. And my Benedict solution in my starch. All right, so as you can see, nothing has happened yet. That's because we need to add our Benedict solution to warm water. So we're going to do a little turn here. I don't know if that's going to work out too well. All right. So I've got my water going. Oh my gosh. So let's see if this works. I thought this pot would be high enough, but it's not. So let's let that sit in the hot water for a second. By the way, guys, you shouldn't be trying any of these things at home at all. That's why I'm doing them on video for you. Okay. All right. And let me see if I can pick this up and bring this over there. We can see, if we look in here, we see that there is a color change occurring in our glucose test tube. Right, I'm gonna pull those out in a second. So just give me 1 second, stand by. All right, so let me pull these out here. All right, so these are very hot, so pardon my mitts here. So here's our water test tube, still blue after being in the heat bath. Okay. And then I have our starch and our glucose. Ooh. Oh. Oh is correct. All right. So we did have an anomaly happen. I don't know what happened, maybe because I messed up my test tubes, but here's our glucose test tube, and there was a color change. That is because there's glucose present in this test tube, right? So it did turn into an orange color. And then our starch one did a little something different here. So there might have been some sugar, some glucose in this tube, or I don't know, I might have messed up my pipettes, but our starch tube should have stayed blue. It's kind of a green color right now. Generally, if we were to do this in the lab, we would have rerun this test, but since we're doing this on video, we're going to pretend like this didn't happen, right? We're going to pretend like this is blue, because I probably either contaminated my test tube or I contaminated my pipette somehow. It happens in science. All right, let's go back to our chart here, right? So now we're working on this part of the chart where we just tested our Benedict solution. So our distilled water, I'm not going to touch them because they're still hot, is blue. No color change, which has indicated that there is no glucose in that test tube. Our starch test tube should have also stayed blue, so we're going to write blue because there's starch in there, not glucose. And then our glucose test tube with our Benedict solution at a little heat, and what color did we get? We got an orange color. So the choices you can write for this color, because the Regents likes to be word specific, is orange, rust, or red orange. Those are the 3 acceptable colors for this test, okay? All right. So now we have done the chemical testing, which is the part 2. Well, not really the part 2. Part 1, second part of part 1. All right. So then we have a question down here. What test would you need to perform to prove that it is a combination of glucose and the glucose indicator solution that changes color when heated, and not just the glucose or the glucose indicator solution alone? So what could we do here to answer this question? Well, is the combination of glucose and the glucose indicator, because if we boiled if I took a test tube of just this and boiled it, which is essentially what we did here with the water, it would still stay blue, right? So To test that it's the combination of glucose and glucose indicator solution, we would test just the solution. And then we would test just glucose. And then test both together. Which is basically what we did here, right? We did the Benedict solution in just water, and then we did the Benedict solution in glucose. So we pretty much basically just proved that it is the combination of the glucose solution and the glucose indicator that gives us the positive for glucose. All right, so let's see. We need to go back to our cell here. So let's take a look. Did we have a bit of a color change in our cell? Looks like we did, right? So this is not what our cell looked like when we started. We have a bit of a reaction going on here. Look at that. All right. So if we go back to page 4 of your packet, we are going to examine our cell in our beaker, which we did have a color change, right? So what did we observe here? So we're going to record our changes. So the cell is a blue, and the beaker okay, so those are basically what we got here, right? The cell turned to a blue-black color, as we can see here. This is not what we started with. And then the beaker is still an amber color. So let's move down on here. So our final state, based on what we did just now with our chemical testing, what caused the reaction inside of the cell? What must have moved, right? So we started with starch and glucose inside the cell, as we labeled on our initial diagram, and iodine outside of the cell. So what happened in our final state here? Well, we have starch and iodine inside the cell, maybe a couple of glucose, right? So that's where we're at so far, and we still have iodine out into the beaker. So if the iodine moved in, do we think that something might have moved out of the cell and into the beaker? How can we test for that? So basically what we're going to do is I'm going to take this one here. I'm going to dump this out, clean it real quick. Right, so I have my empty test tube that I just used, and I'm going to take some of the solution from the beaker, this amber colored solution right here, and I'm going to put it into our test tube. And if I'm testing for glucose, I'm going to put some of the Benedict solution into there as well. And I'm going to pop it in the heat bath real quick. So I'm going to put some of that in there, right? Because we were looking for glucose. See if glucose has moved into or has moved out of the cell. I'm going to place it into our heat bath right here for a minute and see what we get. All right. So while we wait for that to heat up for a second, we can work on question number 1 here. What is the best explanation for the color change that occurred inside the cell? Well, we want to use specific vocabulary here. What is this entire lab about? It's about diffusion. So diffusion should probably be in your answer, right? So the iodine diffused into the cell. Does that make sense, right? Iodine diffused into the cell. We learned from our chemical testing that the way we can test for starch is with iodine, and we get a color change, which is what we got here, right? We got our color change in our cell. Did any starch diffuse out of the cell? Explain how you can tell. Well, if starch diffused out of the cell, what color would our beaker be? Our beaker would be a blue-black color, right? Our beaker is still amber colored. So I'm going to go with no. Starch did not diffuse out of the cell. And explain how we can tell. There was no color change. The beaker is amber colored. If starch diffused out of the cell, the beaker would be right. So I hope you guys see that, so that we know that iodine has diffused into the cell because inside the cell it's blue and black colored, but starch is not diffused out of the cell because the beaker is still amber colored, right? Okay, did any glucose diffuse out of the cell? That's what we're working on right now, and I'm waiting for our heat bath to do something. All right, so I'm just taking a look here. All right, so I turned the water off, so it's a little slow going here. We're getting the burner going again to make it warm enough. So bear with me here. Let's see if we can move on to some other questions. All right. So we can kind of skip here while we wait. Oh, actually, we don't have to wait anymore because if we take a look, this is our test tube from the sample that I took from the beaker water, right? So we got a color change. We got an orange color change, which means that glucose did diffuse out of the cell, right? So we're going to put, for question number 3 on page 4, we're going to put yes. Glucose did diffuse out of the cell, and we can say that we tested it with Benedict solution. We took a sample of the beaker liquid and tested positive for. So if we go back up here to our final state diagram, we can also add some G's outside of the cell where those I's are, right? Because we have determined that glucose diffused out of the cell, right? By testing that with the Benedict solution. Okay, we have a positive Benedict solution test, orange color for glucose, all right? So then we just have 4 more questions to work on regarding this part. And then that is all I have for you guys for today. I will work on doing part 2 of this lab on video for you with the onion cell, where we watch osmosis happen through the onion cell. I do have a microscope here at home. It's just getting that set up so that you guys can actually see through the microscope. That is what I'm trying to troubleshoot right now. So stay tuned for part 2. I will be working on that. I do have to go to the grocery store to pick up an onion because, well, we don't have red onions in my house, and the grocery store is kind of a scary place right now. So as soon as I muster up the courage to venture out there, we will work on part 2. All right, so question number 4 on page 5. What substances diffused through the membrane? Right, so iodine, right? Iodine diffused through the membrane. That's how we we're able to get this color reaction, the color change in our cell, but we can't really see too well right now, right? That's how we're able to get our color change in our cell. And then based on our test here with our beaker water, we got our positive result for glucose. So glucose diffused through the membrane, right? Which substances did not diffuse through the membrane? Well, we know starch didn't, because if starch did, we would have a blue-black color in our beaker, so starch, right? And explain why some substances were able to pass through the membrane while others were not able to. This goes back to our diffusion and knowing about our cell membrane. Our cell membrane is a semi-permeable membrane, which means, or a selectively permeable membrane, which means that it is selective about what it allows into and out of the cell. I did review with you guys a little bit about diffusion before we broke off for our social distancing. And basically, why were they not able to pass through the cell? Well, starch and glucose and iodine are all different size molecules, right? Glucose and iodine are smaller than starch, which is why the glucose and iodine were able to diffuse out of the cell versus the starch, which did not diffuse out of the cell. So it's safe to say that we have a couple answers we could use here. Starch molecules were too big, too large, and selectively permeable membrane. And what we know about selectively permeable membranes is they are picky. They only allow certain things in and out. Okay. All right, and then our last question, number 7 on page 5. In the initial state diagram below, starch indicator solution is indicated with the letter I because it contains iodine. Using the letters S for starch and G for glucose indicate the areas where each of these molecules are located in both diagrams. Be sure you indicate the location of iodine molecules in the final state diagram too. So this is basically what we did on page 4. Right, so we started with iodine out into the beaker, right? We have iodine, all I's, all the way out in the beaker. And we had S's and G's in the cell, right? Our starch and glucose in the cell. Our final state, we had iodine still outside of the cell, right? We also had glucose outside of the cell, based off of our chemical indicator test of our beaker solution, right, that we did here with our orange solution. And then in here, we had starch and iodine, right? You can also put a couple G's in there as well, because I doubt all of the glucose left the cell. But for the most part, you should have starch and iodine in the cell and glucose and iodine outside of the cell in your final state, all right? All right, guys, so that is it for Miss Patterson's Kitchen Science for today. Again, I will work on getting the part 2 of this lab up and done for you guys so you can see these concepts. If you have any questions, you guys have figured out how to chat with me on Teams or email me. And again, don't worry about the due dates for the work. As long as you get the majority of your stuff done by April 1st and you're showing me some sort of effort, you will get the credit. So just keep working hard. Keep occupying yourselves, guys, and stay safe and stay at home with your families. And we'll see you guys soon. Have a good day, guys.